Earth hasn't always been a planet of forests, dinosaurs, and humans. For most of its history, it was a quiet, alien world. Wrapped in oceans and coated in a thin living film, this slime was made of simple, microscopic life forms that ruled the planet for billions of years. They shaped the air we breathe, the rocks beneath our feet, and even the future of every living thing that would one day walk, swim, or fly. Below are 10 chapters you can use to structure your video about the time Earth was covered in living slime. Long before the first dinosaur took a step, before the first fish wriggled through the water, or the first tree reached for the sky, Earth was almost unrecognizable. If you could travel back in time more than 4 billion years, you wouldn't see a blue and green planet full of life. You'd see a world that looked wild and hostile, yet quietly preparing the stage for everything that would one day be alive. The continents as we know them didn't really exist yet. Instead, the surface was a patchwork of forming crust. Jagged, black volcanic rock, constantly being cracked, melted and rebuilt from within. Volcanoes were everywhere, rising like open wounds from the ground and from the seafloor, spewing out lava, ash, and clouds of gas. Rivers of molten rock flowed across barren plains and into early oceans, turning the water hot, murky, and full of dissolved minerals. Above this restless landscape, the sky was thick and strange. There was no blue, no white clouds drifting lazily overhead. The atmosphere was filled with volcanic gases, carbon dioxide, methane, ammonia, nitrogen, water vapor, and almost no oxygen at all. The air would have been toxic to us, burning the lungs of any modern animal that tried to breathe it. The sun, younger and slightly dimmer than it is today, shone through a hazy, reddish-brown sky. Without an ozone layer to filter ultraviolet radiation, the sunlight that reached the surface was harsh and dangerous. Meteorites still rained down from space, leftovers from the formation of the solar system. Some were small rocks that burned up in the atmosphere, but others were enormous, striking the planet with enough force to shake the crust and boil parts of the ocean. Each impact was a disaster and a gift at the same time, a burst of destruction and a fresh delivery of new minerals and chemical ingredients from space. Yet despite how violent it was, Earth was cooling. The oceans were forming. At first, the surface had been an ocean of magma, but as millions of years passed, the outer layer solidified into a crust water, released from inside the planet through volcanic eruptions and delivered by icy comets and asteroids, began to collect on the surface. At first, it was just rain hissing and steaming as it hit hot rock, but slowly, puddles turned into lakes. Lakes joined into seas, and seas into vast global oceans. Those early oceans would not have looked like the clear blue water we imagine today. They were dark, cloudy, and rich in dissolved minerals. Iron and other metals washed out of the young crust, turning the waters greenish or even rusty. The waves crashed against bare rock, no beaches, no sand, no plants, no shells. The only sound would have been the wind, the endless roar of volcanism, thunder from lightning storms, and the distant echoes of impacts from the sky. Lightning flashed repeatedly across the sky as thick clouds of water vapor rose from the warm oceans below. Each bolt of lightning was a massive delivery of energy, ripping through the atmosphere and slamming into the surface. At the same time, underwater near the cracks in the seafloor, hot fluids rich in chemicals blasted out of hydrothermal vents, creating sharp temperature differences and strong chemical gradients. In countless locations across the planet, energy and chemistry were colliding over and over again. The day was much shorter than it is now, perhaps only 6 to 10 hours long. Tides, pulled by a moon that was much closer to Earth than today, rose and fell dramatically. Coastlines were constantly flooded and dried as the ocean sloshed back and forth over the crust. In shallow pools along these primitive shores, water evaporated under the raw, unfiltered sun, concentrating whatever chemicals were dissolved in it. From a distance, this young Earth might have looked quiet at times, a smooth ocean, a red sky, a glowing volcanic horizon, but up close, it was a place of endless motion and transformation. Rock, water, air, and energy were constantly interacting. The planet was chaotic, but in that chaos something new was beginning to emerge. At first there was no life to speak of. No cells, no organisms, nothing that could grow, reproduce and evolve. There were only atoms and simple molecules drifting in the oceans and atmosphere. Hydrogen, carbon, nitrogen, oxygen, sulfur, phosphorus. These elements are the basic alphabet of life, but in the beginning they were just scattered letters waiting to be arranged into words. In the warm, mineral-rich waters, especially in shallow pools and around hot vents, these elements started forming more complex molecules. Simple gases like methane and ammonia, dissolved in water and hit by lightning, ultraviolet radiation, and heat, could form organic molecules, carbon-based compounds like amino acids and simple sugars. On their own, these molecules were not alive. They didn't think, they didn't move, and they didn't reproduce, but they were the raw ingredients of life, drifting silently in the ancient seas. Imagine a tide pool along a rocky shore on this early Earth. The water is warm and full of dissolved salts and metals. Volcanic gases bubble up from below. Each morning, the tide washes in new water and new molecules. Each afternoon, the sun beats down, evaporating some of the water and concentrating whatever is left behind over and over. Wave after wave, the pool is stirred, dried, refilled and heated. In this natural laboratory, millions of chemical reactions are happening at once. Molecules bump into each other, break apart, join together, and form new structures. Some of these structures might be long chains of carbon, primitive versions of the molecules that would later become proteins and nucleic acids. Others might be oily droplets that spontaneously form when certain molecules cluster together, creating tiny bubbles with the thin boundary between inside and outside. These droplets are not true cells, but they're starting to look a little like them. Small, rounded compartments that can trap other molecules inside. Deep in the oceans, around hydrothermal vents, another kind of chemistry is happening. Here, superheated, mineral-rich water gushes out from cracks in the seafloor, mixing with colder seawater. The result is a landscape of black chimneys and shimmering towers, surrounded by constantly shifting chemical gradients. In these places, temperature and chemistry change dramatically over very short distances, providing a natural engine for complex reactions. Molecules that would never meet in the open ocean can come together here, driven by flows of heat and energy. We don't know exactly where life first appeared. It might have been in shallow pools along a coastline, in deep-sea hydrothermal vents, or even inside porous rocks where water and minerals could mix in sheltered spaces. But wherever it happened, the general story was similar. Simple molecules began to form more complex ones, and some of those complex molecules developed the remarkable ability to copy themselves. One possible step was the appearance of molecules similar to RNA, which can both store information and participate in chemical reactions. Imagine a strand of such a molecule forming by chance in a favorable environment. If it could help speed up certain chemical reactions, especially ones that led to the creation of more copies of itself, then it would start to behave differently from the rest of the chaotic chemistry around it. It would no longer be just a random molecule, it would be part of a system that could grow. Once copying enters the picture, even imperfectly, something important changes. Evolution can begin. When a molecule copies itself, small mistakes can happen. Some of those mistakes make it worse at surviving, and those versions disappear. But every now and then, a mistake might make the system more stable, more efficient at using resources, or better at enduring harsh conditions. Those versions tend to persist and spread. Over immense stretches of time, these self-copying molecular systems became more organized. Some became trapped inside tiny droplets or membranes, formed from simple fats and oils, creating primitive protocells. Inside these little bubbles, chemistry could be controlled and concentrated, separate from the outside environment. The boundary of the bubble protected its contents, while still allowing certain molecules to pass in and out. With the appearance of these protocells, the line between non-living chemistry and living cell began to blur. At some point, after countless cycles of trial and error on a planetary scale, one of these protocells crossed a threshold. It became capable of doing three crucial things: maintaining its internal chemistry, using energy from its surroundings, and reliably making copies of itself. It might still have been incredibly simple, nothing like a modern bacterium, but it was alive in the most basic sense. This first true cell, or the first group of such cells, would have been microscopic and invisible to the naked eye. It did not know that it was starting a story that would lead to every plant, every animal, every fungus, and eventually to us. It was simply following the rules of chemistry and physics in a very particular way, repairing itself, feeding, and dividing. From that moment on, Earth was no longer just a planet of rock, water, and air. It had something new, a thin spark of life spreading through its oceans. These earliest cells floated in the ancient seas, feeding on chemical energy and multiplying slowly. They had no eyes, no brains, and no awareness, but they had the one thing that would shape the future of the planet: the ability to persist, to adapt, and to change over time. Billions of years later, we look back and call this moment the origin of life, as if it were a single event. In reality, it was probably a long, messy transition filled with many failures and many dead ends. But eventually the successful versions won out. The oceans that once held only random, lifeless molecules now contained living, self-sustaining cells. These tiny, fragile forms of life didn't know it, but they were the ancestors of everything. The age of pure chemistry was ending. The age of biology, the age of living slime, had begun. As those first tiny cells continued to divide and spread through the ancient oceans, something subtle but important began to change. Life was no longer just a scattered presence, one lonely cell drifting here, another over there. In certain places where conditions were just right, the microbes began to gather. They clustered on rocks, they clung to grains of sand, and they formed thin, slippery films on the seafloor. Over time, these films thickened, layer upon layer, until huge stretches of the shallow seas were covered in soft, squishy blankets of life. If you could dive into those early oceans, you wouldn't see fish or seaweed. Instead, you'd see vast plains of living slime. In shallow, sunlit waters, carpets of microbes spread like biological paint, covering almost every surface they could hold on to. Each carpet was a tightly packed community of cells, stacked in layers, all pressed together so closely that a single square centimetre might contain billions of tiny organisms. These early microbial mats were not just random clumps, they were structured. At the very top, facing the light, lived microbes that needed sunlight or oxygen. If any was available at all, just below them are microbes that preferred dimmer conditions, feeding on substances produced by the layer above. Deeper still, where almost no light could reach and little oxygen was present, lived microbes that survived on different chemical reactions, breaking down waste products and recycling nutrients back into forms that the upper layers could use again. It was like a layered city made entirely of bacteria. The top layer was the bustling surface district, where energy from the sun or from dissolved chemicals first entered the system. Below that lay the middle neighborhoods, where byproducts and leftovers were transformed into something useful. At the very bottom were the basement levels, where decomposers quietly broke down whatever drifted down from above. Every layer depended on the others, and together they created a self-sustaining system. These mats were tough too. Waves could crash over them, tides could rise and fall, and sediments could rain down from the water above. But the mats clung stubbornly to the rocks and mud, held together by sticky substances secreted by the microbes themselves. They formed a gel-like matrix that protected the cells inside. Dirt and sand might settle onto them, but the microbes didn't simply give up. They grew upward through the debris, forming new layers on top, slowly building structures that could last for thousands of years. In some places, these microbial carpets did something even more impressive. They started to build stone. As the mats trapped sediment and minerals, they gradually formed rounded, layered mounds called stromatolites. Imagine a low, dome-shaped rock rising from the shallow seafloor, made not by wind or waves, but by countless generations of microbes living, growing, and trapping particles. Each thin band inside the stromatolite marked an old microbial surface, buried and preserved as the mat grew upward toward the light. Day after day, as the tide came in, new sediments washed over the mats. Night after night, as the tide went out, the microbes continued their quiet work. Over centuries, these cycles left behind rock records of living slime, a kind of fossilized biography written in layers of stone. Some of the oldest fossils we have today are ancient stromatolites, silent witnesses to a time when slimy microbial communities were the masters of the planet. For billions of years, these mats dominated Earth's shallow seas. Wherever there was light, water, and a solid surface, they spread. Along coastlines, across tidal flats, around volcanic islands, and on submerged shelves of early continents, they shaped the environment around them, stabilizing sediments, changing the chemistry of seawater, and gradually influencing the atmosphere above. Life at this point was still entirely microscopic, but it was no longer simple. Within the mats, competition and cooperation played out constantly. Different types of microbes battled for space, light, and nutrients. Some produced substances that discouraged rivals. Others formed partnerships, exchanging molecules in a kind of chemical trade. If one layer of the mat was damaged by drying, burial, or predators, the surrounding microbes could slowly repair the wound, regrowing into the empty space. From above, this world might still have looked barren, rugged coastlines, empty skies, no plants or animals. But just below the water's surface, an enormous living skin coated the planet Earth, was turning into a world wrapped in biofilm, where slimy communities of microbes were quietly rewriting the rules of the environment. Their influence was about to grow even more dramatic, because among them was a group of tiny organisms that had discovered a revolutionary trick for using the sun. In the upper layers of some microbial mats lived a special kind of microbe that could take sunlight itself and turn it into energy in a very efficient way. Instead of relying only on chemical energy from the environment, These microbes began to use light as their main power source. They developed a complex molecular machine, pigments and proteins that could capture photons from the sun and use that energy to drive chemical reactions inside the cell. These microbes are what we now call cyanobacteria, often referred to as blue-green algae, even though they are not true algae at all. They contained pigments that gave them a bluish or greenish colour, letting them soak up sunlight in shallow water. Using that light energy, they pulled in carbon dioxide from the water and combined it with hydrogen taken from water molecules to make sugars, stored fuel they could use to grow, repair themselves, and divide. But this process had a dramatic side effect. It released oxygen. To us, oxygen is the gas we need to breathe. To most early life on Earth, it was something entirely different. A reactive, dangerous chemical that could break apart delicate molecules and damage cells. When cyanobacteria first began releasing oxygen as waste, it didn't immediately fill the air. At first, it reacted with anything it could find in the oceans. One of the main things it reacted with was iron. Early Earth's oceans were rich in dissolved iron, carried by rivers from the young crust and left floating invisibly in the water. When oxygen met this dissolved iron, it oxidized it, essentially rusted it, turning it into solid particles of iron oxide that sank to the sea floor. Layer by layer, this process built up enormous deposits of banded iron formations, striped rocks made of alternating layers of iron-rich minerals and other sediments. For a long time, this chemical reaction soaked up most of the oxygen produced by the cyanobacteria. The oceans acted like a sponge, absorbing and locking away oxygen in the form of minerals. To the naked eye, not much might have seemed different. The sky still had no blue tint, the air was still poisonous to modern life, and the microbial mats continued their daily routines. But under the surface, a revolution was underway. The cyanobacteria thrived. With sunlight pouring down from above and carbon dioxide abundant in the air and water, they had access to nearly limitless energy. Microbial mats that contained these photosynthetic bacteria could grow thicker and spread farther. Wherever shallow, sunlit water was present, cyanobacteria multiplied, forming new mats and reinforcing old ones. Slowly, the ocean's iron sponge began to fill up. Eventually, there was less and less dissolved iron available to capture the oxygen. Once the iron was mostly locked away on the seafloor as solid minerals, the extra oxygen had nowhere else to go. It started to build up, first in the oceans, and then, gradually in the atmosphere. To the microbes that could tolerate or use oxygen, this was an opportunity. Oxygen-based chemistry can release far more energy from food molecules than earlier forms of metabolism. Any organism that could figure out how to handle oxygen safely and use it for respiration would have a powerful advantage. But for many of the existing microbes, oxygen was a crisis. It attacked their cell membranes, damaged their molecules, and disrupted their way of life. What followed was one of the most dramatic turning points in Earth's history, often called the Great Oxygenation Event. Over millions of years, oxygen levels crept upward. At first, the change might have been subtle, noticed only by the microbes themselves as the chemistry of their environment shifted. But as oxygen continued to rise, entire ecosystems of anaerobic, oxygen-hating organisms were pushed back into the shadows, into deep mud, underground, or into environments where oxygen still couldn't reach. From the point of view of those early anaerobic life forms, it was like a slowly spreading poison. Waters that had once been safe became hostile. Places where they had thrived were taken over by mats of cyanobacteria and other microbes that could cope with oxygen or even benefit from it. This wasn't a sudden, world-ending explosion, but a long and relentless wave of change that swept across the planet's seas. At the same time, oxygen in the upper atmosphere began to form ozone, a special form of oxygen that collects in a thin layer high above the surface. This ozone layer started to block some of the sun's harsh ultraviolet radiation. Ironically, the gas that was deadly to many early microbes also set the stage for life to eventually leave the safety of the oceans and venture onto land, protected from radiation by a shield of its own waste. The cyanobacteria, meanwhile, continued their quiet work, unaware of their planetary scale impact. Each cell simply absorbed light, took in carbon dioxide and water, and produced sugar and oxygen. Trillions upon trillions of these small, simple organisms collectively transformed the atmosphere. They turned Earth from a world almost entirely without oxygen into one where oxygen became a dominant gas. This transformation was not quick. It took hundreds of millions of years for oxygen levels to rise from nearly zero to something significant. Along the way, the composition of the atmosphere shifted. Climate patterns changed and new kinds of minerals formed. The very colour of the sky and the chemistry of the oceans were altered by this invisible army of photosynthetic slime. In shallow bays and tidal flats, you could have seen the source of this change if you looked closely enough. Broad, wrinkled mats of cyanobacteria rising and falling with the tides. During the day, tiny bubbles of oxygen would have formed within the slime as photosynthesis ramped up, sometimes escaping to the surface as delicate streams of gas. At night, when the light faded, the mats would switch to other ways of getting energy, resting until sunrise brought power back to their molecular machinery. These carpets of living slime were the first great planetary engineers. Without any intention or plan, they restructured the atmosphere, rewrote the surface chemistry of the planet, and laid down mineral deposits that we would one day mine as iron ore. They also made possible the evolution of more complex life forms that could use oxygen for respiration, unlocking a path toward larger bodies, more active lifestyles, and eventually the rich tapestry of plants and animals that would cover the earth. Long before forests appeared, before insects buzzed in the air, and before animals swam in the seas, the world belonged to the microbes, and especially to the sun-harvesting cyanobacteria. They turned sunlight into sugar, sugar into growth, and growth into a global transformation. The age of the living slime was not just a strange chapter in Earth's past. It was the foundation upon which every later chapter was built. For a long time, the oxygen made by photosynthetic slime never got very far. It was immediately swallowed up by the surrounding environment, used up by chemical reactions with iron, sulfur, and other elements dissolved in the oceans. The world was like a huge chemical sponge, soaking up every bit of oxygen that cyanobacteria released. But that couldn't last forever. Gradually the balance began to shift. The ocean started running out of things that could easily react with oxygen. A lot of the dissolved iron had already rusted and sunk to the seafloor as solid minerals. Vast layers of iron-rich rock were quietly laid down, forming thick bands that would one day be mined by humans. But for now, they were just part of the slow, deep memory of the planet. As the chemical sponge of the oceans filled up, the oxygen from the slime mats began to spill over. Bit by bit it started to remain dissolved in the seawater instead of being instantly consumed. From there, some of it diffused into the air above. At first, the amount of oxygen in the atmosphere was still tiny. Far too little for any animal like us. But compared to the previous near-zero levels, it was a huge change. To many of the microbes that had evolved in the old oxygen-free world, this change was catastrophic. Oxygen is a hungry, reactive molecule. It tears electrons away from other molecules and can damage proteins, membranes, and genetic material. For organisms not prepared to deal with it, oxygen was not a blessing. It was a toxin. Imagine living in a world where the air and water themselves are slowly becoming poisonous. That was the reality for countless anaerobic microbes, organisms that not only didn't need oxygen but were harmed by it. They had evolved in a gentle chemical environment, one where different gases like methane, hydrogen, sulfide, and carbon dioxide supported their metabolism. Oxygen entering this world was like acid slowly being poured into a delicate system. As oxygen levels crept up, entire habitats changed. Shallow, sunlit waters where cyanobacteria thrived became increasingly hostile to anaerobic microbes. Places that had once been safe and productive began to corrode their very cells. Some of these microbes migrated deeper into the sediments where oxygen couldn't reach. Others moved into environments that remained protected, deep underwater, inside rocks, in mud at the bottom of lakes and seas, and in hot oxygen-free vents. But many simply vanished, wiped out by a gas that today we think of as fresh air. This slow global shift is sometimes called the first great mass extinction. It didn't happen in an afternoon. It took millions upon millions of years, but on a planetary scale it was sudden. Our whole style of life, an oxygen-free way of living, was pushed into the corners and shadows. Meanwhile, life that could cope with oxygen had an opportunity. Some microbes evolved enzymes and protective molecules that could neutralize oxygen's harmful effects. They developed antioxidant systems, tiny molecular shields and tools that allowed them to live in places where oxygen was present, without falling apart. Others did more than just tolerate oxygen. They began to use it. Using oxygen to extract energy from food molecules is incredibly efficient. Compared to earlier, anaerobic metabolisms, oxygen-based respiration can release much more energy from the same amount of nutrients. Microbes that learned how to breathe oxygen, in a sense, gained access to a powerful new fuel system. This extra energy would later make larger, more complex forms of life possible. But in the beginning, it simply meant that some bacteria could outcompete others, grow faster, and survive in the changing environment. But oxygen didn't just affect living things, it also changed the climate. Before oxygen rose, the atmosphere contained a lot more methane, a greenhouse gas far more potent than carbon dioxide. Methane helped keep the early Earth warm, trapping heat from the young, dimmer sun. However, when oxygen began to accumulate, it reacted with methane, breaking it down and converting it into carbon dioxide and water, gases that trap less heat. As methane levels dropped, the planet began to cool. What started as a chemical side effect of the slime's photosynthesis slowly turned into a shift in global temperature. With less methane to hold in heat, more of the sun's energy bounced back into space. Over time, this cooling intensified. Ice that formed at the poles lasted longer. Snow that fell in colder regions didn't melt as quickly. Ice and snow are bright. They reflect sunlight back into space better than dark oceans or bare rock. So as the planet cooled and ice spread, more and more of the sun's energy was reflected away. This created a feedback loop. More ice means more cooling, which means more ice, which means even more cooling. Eventually, this process may have gone to an extreme. Earth entered one of the coldest phases in its entire history, a time when thick sheets of ice may have stretched from the poles almost all the way to the equator. Seas froze over, continents were buried under glaciers, and the planet became, in many scientists' view, something close to a snowball Earth. At first glance, this sounds like the end of the story. A frozen planet covered in ice seems like a death sentence for life, especially fragile microbial slime that had once basked in shallow sunlit seas. How could these communities survive when their warm, watery homes were capped by kilometers of ice? Yet life did not disappear. These slime adapted, retreated, and found refuge in hidden places. Even during these extreme glacial periods, Earth was not a perfectly smooth, white marble. Near the equator, some areas of open water may have remained, slushy patches of ocean where ice was thinner or cracked. Around active volcanoes, heat from below kept water liquid beneath the ice, or created small areas where the surface melted. Along tectonic plate boundaries, hydrothermal vents continued to pump hot, mineral-rich water into the deep sea, completely sheltered from the frozen surface. In all of these places, microbial life found ways to endure. Deep under the ice, hydrothermal vent communities carried on almost unchanged. These ecosystems didn't rely on sunlight in the first place. Instead, they drew energy from chemicals rising from beneath the Earth's crust. For them, the frozen surface oceans were almost irrelevant. Their world was one of darkness and heat, where bacteria and archaea used sulfur or other chemicals as fuel, forming the base of rich microbial networks. Closer to the surface, the situation was more precarious. Some microbes survived in very thin films of liquid water that existed even on or within the ice itself. When sunlight hits dirty or dusty ice, it can melt tiny pockets, creating little pools of liquid called cryoconite holes. These are like miniature ponds on the surface of glaciers, where dust, ash, and microorganisms collect. Even in a snowball Earth scenario, regions exposed to sunlight could have hosted these microhabitats, tiny oases of liquid in a frozen desert. Within cracks and fractures in the ice, where pressure and geothermal heat cooperated, thin veins of unfrozen water could persist. Microbes could live there, using dissolved nutrients carried by slow, creeping flows. On the seafloor beneath thick ice, microbial mats may have continued to grow, powered by faint filtered light or by chemical energy from below. Life shrank back into the cracks and corners, but it did not vanish. Some of the microbial mats that once coated wide, sunlit shallows now clung to rocky outcrops in patches of open water, perhaps near volcanically active islands that poked up through the ice, or in equatorial belts where conditions were slightly milder. Cyanobacteria, those same slime makers that had helped trigger the cooling, persisted wherever light and liquid water still overlapped. Their photosynthesis slowed but did not stop. Inside sediments, buried beneath layers of mud and ice, dormant cells waited out the worst conditions. Microorganisms can enter states of extremely low activity, almost like sleep, when conditions are too harsh for growth. They can survive long periods of cold, starvation, or dryness in this suspended state. When conditions improve, they wake up and begin dividing again. During Snowball Earth episodes, countless microbes may have endured like this, scattered like tiny seeds of life across the planet. All the while, the same forces that had plunged Earth into deep freeze continued to operate, just in reverse. Volcanoes never stopped erupting. With much of the surface locked under ice, weathering of rocks slowed dramatically. Under normal conditions, weathering removes carbon dioxide from the atmosphere as rainwater reacts with rocks. But under global ice, that process was greatly reduced. Volcanic activity, however, kept adding carbon dioxide to the air. Because the carbon dioxide being added wasn't being efficiently removed, it began to build up. Slowly, the greenhouse effect strengthened again. The atmosphere trapped more heat. Over millions of years, this accumulation of carbon dioxide warmed the planet enough to begin melting the global ice cover. At first, cracks formed. Then, patches of open water appeared, especially in equatorial regions. As more dark ocean surface was exposed, it absorbed more sunlight, amplifying the warming. From the perspective of the frozen world, this must have been a slow, planet-wide thaw, even though no one was there to see it. Glaciers retreated, ice thinned, and the heavy white shell covering the oceans fractured and shrank back toward the poles. Rivers of meltwater poured off the ice-covered continents into the sea, changing salinity and circulation patterns. Torrents of liquid water carved fresh scars into the crust. When the ice finally pulled back, life rushed into the newly opened spaces. Microbial mats that had been confined to tiny refugees suddenly had entire coastlines to colonize again. Cyanobacteria spread over bare rocks, shallow seas, and tidal flats, just as they had done in the ages before the freeze. Their greenish-blue films once again covered freshly exposed surfaces like paint, kick-starting photosynthesis in newly sunlit waters. It's possible that the extreme conditions of Snowball Earth helped drive evolution forward. Surviving such a harsh climate would have required resilience, flexibility, and new strategies for endurance. Populations that made it through the freeze might have emerged more diverse, or with new genetic combinations that allowed them to exploit changing conditions more efficiently. During and after these frozen episodes, the chemistry of the oceans shifted dramatically. As glaciers melted, they released enormous amounts of minerals and nutrients that had been locked in ice and rock. These nutrients washed into the seas, feeding explosive blooms of microbial growth. Sudden surges of life could have changed oxygen levels, carbon cycles, and the balance of different microbial communities all over again. So you have this strange, almost paradoxical picture. Earth, covered in ice, yet still alive. A frozen world where inside cracks in ice, at the bottom of the sea, around smoky underwater vents, and on sunlit patches of meltwater, slime persisted. The same living slime that had poisoned the old atmosphere with oxygen and helped trigger global cooling also weathered the crisis it created. By the time the great ice sheets retreated and the planet warmed again, life had proven something important about itself. It could survive both heat and cold, abundance and scarcity, darkness and light. The age of slime did not end in the freeze. Instead, it passed through fire and ice, oxygen and poison, chaos and calm, and kept going. When the Ice Age finally loosened its grip and oceans opened wide under a clearer, oxygen-rich sky, Earth was left transformed. The atmosphere now held far more oxygen than it ever had before. The climate, though still changeable, was no longer dominated by the same thick methane blanket of its youth. The crust bore scars of ice and floods, and everywhere in fresh seas and along new coasts, microbial life was ready to build again. From these survivors, from mats that never fully died, from vent communities that never froze, from cells that waited patiently inside rock and sediment, the next chapter of Earth's story would unfold. The world was still ruled by slime, but it was no longer only building a chemical foundation. It was now preparing slowly and silently for the rise of something far more complex than anything that had existed before. Within those vast microbial mats that carpeted ancient shorelines and shallow seas, life began to do something new. It started to act together. At first glance, a microbial mat might look like just a sheet of slime, but if you could shrink yourself down to the scale of a bacterium and step inside, you'd discover a world buzzing with structure and purpose. Each mat was a layered, crowded metropolis made of cells instead of people, chemistry instead of machines. Near the top, in the thin zone where sunlight could still reach, lived the sun harvesters, cyanobacteria and other photosynthesizing microbes. They were like the farmers and power plants of this tiny world, absorbing light and turning it into chemical energy, making sugars and organic molecules that other organisms further down could eventually use. Just beneath them lived microbes that were better suited to dim conditions. They fed on the waste and leftovers from the upper layer, organic molecules that drifted downward, fragments of dead cells, and other byproducts of photosynthesis. These middle microbes were recyclers and processors, transforming one kind of molecule into another and passing energy deeper into the mat. Further down, where little or no oxygen could reach, lived yet another set of specialists. These cells fed on substances that would be toxic to many others: hydrogen sulfide, methane, or other reduced chemicals. They could strip away electrons, release energy, and excrete new compounds that diffused upward or sideways, becoming food for yet another group. Nothing was really waste in this world. Almost everything could be used by someone. Holding this whole community together was a sticky gel-like material secreted by the microbes themselves. It was made of sugars, proteins, and other molecules, like a biological glue. This matrix protected the cells from drying out during low tides, shielded them from ultraviolet radiation, and helped trap nutrients and minerals. It turned the mat from a loose crowd of microbes into a coherent structure, a slime city with walls and streets made of chemistry. Inside this structure, the microbes didn't just sit passively. They communicated, though not with voices or thoughts, but with molecules. Cells release small chemical signals into their surroundings, messages that diffuse through the matrix. When the concentration of these signals reached a certain level, it told the microbes something important. There are many of us here. The community is dense. This type of communication, often called quorum sensing, allowed microbes to change their behavior depending on how crowded their environment was. When they sensed many neighbors, they might switch on genes that helped them cooperate, producing more matrix to thicken the slime, making enzymes that digested complex food that no single cell could handle alone, or releasing substances that protected the group against harmful chemicals and invaders. Different layers of the mat could talk to each other in this way. The top layer might respond to strong sunlight by increasing its production of protective pigments, shading the layers below. The bottom layers might adjust their metabolism depending on how much oxygen and organic material drifted down from above. Over time, these coordinated responses turned the mat into a dynamic system, capable of adjusting to changes in light, temperature, and nutrients. The structure itself shaped the environment. As mats formed thicker and thicker blankets over sediments, they slowed down water flow near the seafloor. Tiny particles carried by the current settled gently into the slime and were trapped there. Minerals dissolved in the water reacted with substances produced by the microbes and precipitated out, gradually forming solid layers. The mat's own waste products helped build a mineral skeleton beneath it. In some places, this process created stromatolites, layered rock structures that rose like low domes or mounds from the seafloor. You could think of them as the skyscrapers of slime cities, built one microscopic layer at a time. Each new generation of microbes colonized the surface, performed its metabolic work, trapped particles, and was eventually entombed under the next generation's layer. From the outside, they looked like lumps of rock. Inside, they were the fossilized remains of countless ancient communities. Even within a single mat there was diversity. Not all microbes did the same job. Some were specialists at fixing nitrogen from the atmosphere, turning inert gas into usable nutrient forms. Others were experts at breaking down complex organic molecules into simpler pieces. Still others could switch between modes of metabolism, depending on whether light or certain chemicals were available. This division of labor made the community as a whole more resilient. If conditions in the environment shifted, say the water became more saline, or a volcanic eruption changed the chemical balance, some types of microbes might struggle, but others were ready to take over. The makeup of the mat changed, but the mat itself survived. It was like a city changing its population and industries over time, while still remaining a city. Within these slime cities, genetic information flowed as well. Microbes can exchange bits of DNA between each other, even across species boundaries, through processes like horizontal gene transfer. A useful gene, one that helps a cell resist a toxin, or cope with oxygen, or use a new kind of nutrient, could spread through the community, passing from neighbor to neighbor. The mat functioned almost like a shared library of survival tricks, constantly updating and reorganizing itself as new traits appeared. In some mats, there may even have been primitive electrical activity. Certain bacteria can transfer electrons along chains of cells or through conductive filaments, essentially using the community as a living wire. While invisible to the naked eye at the microscopic scale, these were real currents, tiny flows of charge that allowed microbes to move energy from one part of the mat to another. All of this, communication, division of labor, gene sharing, even electrical signaling, was happening in worlds we would casually describe as just slime. But this slime was not disorganized or mindless. It was a distributed intelligence of chemistry and evolution, where individual cells were parts of a larger self-maintaining system. As these slime cities grew more complex, they created a stable platform where new evolutionary experiments could take place. Cells weren't just struggling alone in a hostile ocean, they were nestled inside communities that buffered them from the harshest changes, recycled their waste and shared resources. In that relative safety, new ways of living could arise, ways that would fundamentally change what it meant to be alive on Earth. One of the most important of these new ways was the idea of one cell living inside another. Sometimes, a larger microbe might engulf a smaller one, not to digest it immediately, but to keep it around as a kind of internal partner, a resident that could perform a useful function. This cooperative arrangement, called endosymbiosis, would eventually give rise to more complex cells with internal compartments, including the ancestors of mitochondria and chloroplasts. But even before that, life inside the mats was moving toward more sophisticated partnerships. Yet not all interactions inside the slime were cooperative. In the same crowded, chemically rich environment where microbes learn to share and communicate, another powerful strategy appeared: predation. For a very long time, life on Earth consisted of relatively simple cells, bacteria and archaea, many of which were tiny and rigid, surrounded by tough walls. They mostly absorbed dissolved nutrients or harvested energy from light and chemicals. If one cell happened to die and rupture nearby, others might benefit by absorbing the molecules released into their surroundings, but it was a passive process. At some point, however, certain cells evolved a different approach. Instead of waiting for food to drift to them, they began to move more actively through their environment. Tiny hair-like structures or whip-like tails, flagella and cilia, allowed them to swim, twist, and glide through the fluids in and around the mats. Mobility opened up new possibilities. Now a cell could leave a depleted area and search for richer chemicals, brighter light, or fresher waste to feed on. With movement came the chance for a new kind of feeding, engulfing. Some larger, more flexible cells developed the ability to extend parts of their membrane around particles, or even other cells, wrapping them up and pulling them inside. At first, this might have been a way to eat clumps of organic debris or mineral-rich particles, but sooner or later, in some quiet corner of a mat, one cell engulfed another living cell and digested it. This was the beginning of predation. From the point of view of the predator cell, it was a powerful innovation. A single bacterial cell is a concentrated packet of nutrients. Proteins, fats, sugars, nucleic acids, and minerals all bundled together. By eating entire cells, a predator could obtain far more energy and raw materials than it might from simply absorbing broken down molecules in the water. It was like switching from sipping soup to eating whole meals. From the point of view of the prey, it was a terrifying development. Suddenly, being small and passive was no longer always an advantage. Areas of the mat that had once been simply crowded now had hidden dangers. Larger motile cells prowling through the matrix, sensing chemical cues and reacting to the presence of potential food. This microscopic arms race began quietly but had immense consequences. Prey cells that happened to have thicker or more rigid walls were harder to engulf and digest. Those that could quickly change direction, hide in narrow spaces, or cling more tightly to the matrix had a better chance of avoiding predators. Any mutation that made a cell less likely to be eaten, by altering its surface, its behavior, or its chemistry, could be strongly favored. Predators in turn evolved sharper tools. Some developed sophisticated ways to latch onto prey, using sticky molecules or mechanical hooks on their surfaces. Others produced enzymes that softened or drilled through protective layers, allowing them to breach cell walls or membranes more easily. Some may have evolved toxins that stunned or killed their prey before ingestion, paralyzing them in place. Over many generations, this back-and-forth pressure pushed both sides toward greater complexity. Predators who were better at hunting and digesting prey survived and reproduced more. Prey that were better at escaping or resisting survived and reproduced more. The result was an escalating cycle of adaptation. This arms race didn't just shape individual species, it reshaped ecosystems. In parts of the mats where predators were common, the community structure might shift, with more resistant microbes dominating. In other regions where predation was rare or environmental conditions favored different metabolisms, the mats might look more like peaceful, cooperative neighborhoods. The patchiness of predator pressure across the slime helped generate diversity, encouraging a wide variety of survival strategies. The emergence of predation may also have encouraged the evolution of larger, more complex cells, the kinds we now call eukaryotes. To hunt effectively, it helped to be bigger than your prey, flexible enough to engulf it, and equipped with internal machinery capable of handling the digestion and management of large meals. This, in turn, favoured cells with internal compartments, stronger skeleton-like structures, and more advanced control over their own shape and movement. Inside some predator cells, not every engulfed microbe was digested. In a few rare cases, an ingested cell might survive and find a way to live inside its host instead of being broken down, if that internal cell could perform a useful function, such as processing oxygen more efficiently or producing food from sunlight. Both host and guest could benefit. Over very long timescales, these relationships transformed from temporary alliances into permanent mergers. The engulfed cells shrank and lost their independence, becoming organelles within the larger cell. These mergers were revolutionary. Cells that contained energy-producing partners, future mitochondria, could generate more power from the same amount of food they could afford to grow larger, build stronger internal scaffolds, and maintain more complex internal chemistry. Cells that hosted photosynthetic partners, future chloroplasts, could turn into tiny self-powered factories, combining the advantages of predation, digestion, and light harvesting. Even before these fully developed eukaryotic cells spread widely, the slime world was already being reshaped by the presence of predators. The mats were no longer just passive carpets, they were arenas. Some areas churned with invisible chases and escapes, slower than any animal hunt we know, but relentless over microscopic distances and long stretches of time. Predators glided between layers, following trails of chemical attractants released by stressed or damaged cells. Prey huddled in denser clusters, benefiting from the safety of the group. Some microbes may have formed thicker biofilms, or retreated to deeper, low-oxygen regions of the mat where certain predators couldn't function well. Others multiplied rapidly, accepting heavy losses to predation, but surviving through sheer numbers, a strategy that remains common among microbes today. Meanwhile, scavengers thrived on the leftovers. Not every attack ended in a clean meal. Some prey cells burst, releasing their contents into the slime nearby microbes, not involved in the original hunt, eagerly absorbed the spilled nutrients. In this way, even failed predation attempts fed the ecosystem, channeling energy and materials into a wider web. From a distance, say, from the vantage point of a human observing billions of years later, it's easy to overlook this entire drama. We see only the fossils of stromatolites, the chemical signatures in ancient rocks, and traces of early oxygen in mineral layers. But woven into that deep history is a story of emergence, cities of slime learning to cooperate, and within them, the first predators learning to hunt. These developments, though microscopic, changed everything. Cooperation built stable, layered communities that could shape their environment, recycle resources, and endure upheaval. Predation lit a new evolutionary fire, driving organisms toward greater size, flexibility, and complexity. Together, they set the stage for a future where the oceans would no longer be ruled only by carpets of slime, but by drifting, swimming, sensing creatures, descendants of both the builders and the hunters that arose within those ancient living films. For billions of years, slime ruled the world as tiny single cells. They formed mats, cities, and invisible empires across the sea floor. But slowly, within this microscopic world, something even stranger began to happen. Life started to build larger bodies, not by making bigger cells, but by joining many cells together into one cooperating whole. The first big step was the rise of more complex cells themselves. Some ancient microbes had already merged with others in partnerships that became permanent. A large flexible cell might engulf a smaller bacterium, but instead of digesting it, the two formed a long-term alliance. The smaller cell provided extra energy or special chemistry, while the larger cell provided shelter and raw materials. Over enormous stretches of time, these swallowed partners turned into mitochondria, the energy factories found in almost every complex cell today. In some lineages, photosynthetic bacteria were also taken inside and eventually became chloroplasts, the light-harvesting engines of algae and plants. These new, complex cells, eukaryotic cells, were like tiny planets with their own internal compartments and machinery. They could handle more complicated chemistry, they could store more DNA and control when different genes were turned on or off. They could grow larger than most bacteria, bend and change shape, and carry out tasks that simpler cells never could. Still, even a complex cell was only one cell. It drifted and swam and fed as an individual. The next leap was for cells to stop living only as separate units, and instead start sticking together, not just as temporary clumps, but as real, organized groups. In the slime world, this wasn't as unlikely as it sounds. Microbes were already living side by side in dense mats, glued together by their own secretions. Sometimes cells divided but didn't fully separate, remaining attached as chains or clusters. Others produced sticky surfaces that made it easier to stay with their neighbours. These loose associations were the beginnings of something new, simple multicellularity. At first, these groups were just shapeless blobs, filaments, or hollow balls made of many similar cells. Being part of a group, however, brought advantages. A cluster of cells was harder for a predator to swallow than a single tiny cell. A larger body could glide more smoothly through water, create its own currents, or sink or float in ways a single cell could not. A bigger surface area could capture more light, or absorb more nutrients from the surroundings. Over time, cells in these groups began to specialize. Some cells might focus on gathering food or light, others might anchor the colony to a surface or help it move. Still others might take on the role of reproduction, dividing to create new colonies while the rest of the group supported them. For specialization to work, cells had to cooperate and give up some of their individual freedom. A cell that spent its existence doing a support job, for example, might not reproduce directly, but its genes could still be passed on through the success of the whole group. This kind of cooperation required new kinds of control. Within multicellular groups, genes started to behave differently. The same DNA was present in every cell, but different parts of that DNA were switched on or off in different places. Some cells became long and thread-like. Others became flat or round. Even in the simplest of these early multicelled organisms, a basic body plan was starting to emerge, controlled by invisible instructions written in molecules and passed down through generations. In the oceans, strange new forms appeared. Soft, jelly-like organisms that had no bones or shells, no eyes or jaws, but were undeniably more than single cells. Some looked like fronds, anchored to the seafloor and waving gently in the currents, drawing nutrients from the water. Others lay flat on the sediment like quilted cushions or discs. Some were shaped like segmented tubes or ovals, with faint ridges running along their bodies. The seafloor was still largely covered with microbial mats, and these new multicellular creatures often lived on top of or alongside them. The mats were like living carpets, and the emerging larger organisms were like the first pieces of furniture placed upon them. Some of these soft-bodied beings grazed on the mats, scraping and absorbing slime as food. Others possibly anchored themselves into or just above the mats, using the stable, sticky surface as a foundation. The interaction between slime and these new bodies was intimate. When a multicellular organism rested on a mat, it blocked light and changed the flow of water, creating little microclimates beneath and around it. The microbes responded, adjusting their growth. In turn, the chemistry of the mats, rich in nutrients and gases, shaped where the multicellular organisms could thrive. The early complex life forms weren't separate from slime. They were growing directly on its scaffold. As oxygen levels continued to rise in the oceans and atmosphere, and as nutrients washed into the seas from weathering continents, it became easier to sustain larger bodies. Oxygen-based respiration allowed cells to extract more energy from their food than ever before, and that extra energy could fuel growth, movement, and maintenance of complicated structures. Slowly, across millions of years, these early multicellular organisms diversified. Some became better at staying fixed in place, evolving broad surfaces to capture light or absorb nutrients. Others became better at moving, sliding, crawling, or pulsing their bodies to navigate their world. Some developed internal spaces, like simple guts, where food could be digested more efficiently. Others experimented with branching forms, tubular shapes, and repeated body segments. The world was still quiet and alien compared to the Earth we know, but it was no longer just a planet of slime and single cells. The mats were now a platform supporting forests of soft fronds, fields of quilted forms, and the first faint stirrings of more active creatures. Slime hadn't disappeared. It had become the ground upon which larger life walked, fed, and evolved. Then something dramatic happened again. This time not a chemical revolution, but a biological explosion. As the conditions in the oceans stabilized after cycles of freezing and thawing, and as oxygen levels rose to new heights, the stage was set for an evolutionary burst. In a span of geologic time that is short compared to the long stillness that came before, the variety of animal life in the seas increased rapidly. This period, known today as the Cambrian explosion, filled the oceans with forms that would have looked shocking compared to the soft, ambiguous shapes of earlier multicellular life. Creatures with skeletons and hard shells appeared. Animals evolved legs, fins, and jointed limbs for crawling and swimming. Eyes developed to see in a way no organism had done before. Mouths with teeth and grasping appendages turned feeding into active hunting. Burrowing animals began to dig into the seafloor, churning and aerating the sediments that had once been undisturbed beneath microbial mats. For the slime world, this was both a threat and a transformation. The once solid carpets of microbes that had blanketed the seafloor for billions of years now faced constant disturbance. Grazers scraped away at the mats, devouring them as a rich, ready-made food source. Burrowers ploughed through the microbial layers, mixing them with sediments and drawing oxygen deep into the mud. Predators chased prey across the seabed, stirring up clouds of particles and tearing up the smooth, slimy surfaces. In many places, the continuous, stable mats that had ruled the shallows were broken apart, replaced by a landscape of mixed sediments, scattered patches of biofilm and active, mobile animals. Microbial life did not vanish. It adapted. Some microbes continued to form mats in protected environments where animals were scarce or conditions were too harsh for complex life. Others retreated deeper into the sediments, forming thin layers out of reach of most grazers. Many microbes found a different strategy entirely. They moved into the bodies of the new animals and plants as symbionts. Inside the guts of early animals, microbes colonized the inner surfaces, helping to break down food and extract nutrients. In return, they received a constant supply of organic material and a relatively stable environment. In this way, animals became walking ecosystems, carrying ancient slime inside them as digestive partners. As plants and algae later colonized land and shallow waters, they also turned to microbes. Certain bacteria formed partnerships with plant roots, helping to fix nitrogen from the air into usable form, while plants provided sugars in return. Fungi, themselves built from threads of microscopic cells, wove around and into plant roots, forming vast underground networks that shuttled water and nutrients from place to place. These networks, too, were extensions of that ancient slime logic. Cooperation, shared resources, and dense living communities. Even deep underground, microbial life spread. Tiny cells infiltrated cracks in rocks, living off the faint energy from chemical reactions between water and minerals far below the surface. Others colonized hot springs, acidic pools, salt-saturated lakes, the deep ocean floor, and the thin films of water trapped in soil grains. Wherever there was even a little energy and a little liquid water, descendants of the first slime could be found. The rise of animals, plants, and fungi did not replace microbial life. It layered on top of it. Every forest, every coral reef, every grassland and swamp that would later appear on Earth depends on invisible microbial communities quietly cycling nutrients and maintaining balance. Microbes fix nitrogen, decompose dead matter, recycle carbon, and shape the chemistry of the air and water. Without them, the larger, more visible forms of life could not survive. Even humans, with our cities, technology, and ideas, are intimately tied to that ancient slime. Our bodies are made of eukaryotic cells whose mitochondria are the descendants of swallowed bacteria. Our own cells still run on the oxygen-rich chemistry that cyanobacteria helped create billions of years ago. Our intestines are home to trillions of microbes that help digest our food, train our immune systems, and even influence our mood and health. Each of us is, in a way, a walking, thinking colony built on microbial partnerships. On our skin, in our mouths, in our lungs, and in our blood, microbes are present in vast numbers. We tend to think of them only when they make us sick, but the majority are harmless or helpful. They are part of our normal state, as natural as our bones and muscles. If you stripped away every one of your human cells and left only the microbial residents, there would still be a ghostly cloud-like outline of a person made of nothing but tiny ancient beings. Even on modern Earth, in a time of forests and mammals and birds and skyscrapers, there are places where the ancient world is still visible. In some shallow, salty bays and lakes, stromatolites still grow today, layered structures built by microbial mats, just as they were billions of years ago. These living fossils are windows into the time when slime cities were the most advanced life on the planet. Look closer at almost any environment and the same story appears. In the ocean, vast blooms of microscopic algae and cyanobacteria drift in the sunlit surface waters, forming the base of marine food webs. In soils, unimaginable numbers of bacteria and fungi thread through the grains, quietly breaking down organic material and feeding plants. In the air, microbes ride dust and water droplets, travelling between continents, in ice, in deep caves, in vents at the bottom of the sea. There are cells carrying on the same fundamental processes that began in the warm pools and dark vents of early Earth. So while we often talk about the time when Earth was covered in living slime, as if it were only a chapter in the distant past, the truth is that this chapter never really ended. The mats may no longer blanket every shallow sea floor, and we now share the planet with forests, insects, reptiles, birds, and humans. But the slime is still here. It has simply diversified, hidden itself, and woven its presence into every corner of the biosphere. It lives in layered biofilms on rocks and teeth, in the coatings on river stones, in the slick algae on pond surfaces, and in the invisible films lining our guts. It shapes the air we breathe, the water we drink, and the soil that feeds our crops. It was the first great shaper of Earth, and it remains the silent engineer beneath almost everything alive. When you step back and look at the story of life on this planet, it's tempting to think that we, with our tools and thoughts, are the main characters. But life began as slime, and slime never left. The towering trees, the flashing fish, the soaring birds, and the thinking humans are all brief experiments built on an ancient foundation of tiny cells that learned how to stick, cooperate, hunt, and transform a hostile world into a living one. In the end, Earth is still a slime world, Only now that slime has grown forests, sculpted animals, and even, somehow, produced a creature capable of looking back through time and telling its story,